So as part of the CCVI, I would really like to welcome Dr. Rob Shave to the University of Guelph. Rob is someone that I think anyone who follows his work will say is quite aspirational in terms of the breadth that he takes. So as a scientist that stays in the lab, I'm always looking fondly at people that get to travel out into real world situations. I don't think my wife would let me do that, but it's really a huge undertaking to do the type of work that you do, Rob, and I think it's extremely powerful, the comparative physiology studies that you do as well. So as an introduction, Dr. Shave is a professor in the School of Health and Exercise Science and Associate Dean Research of the Faculty of Health and Social Development at the University of British Columbia. Dr. Shave has published over 165 research papers, and his research focuses on examining the acute and chronic effects of exercise and environmental stress upon the heart. He uses echocardiography and biomarkers to help provide insight to the beneficial and potentially negative effects of endurance exercise on the heart, and on cardiac remodeling and ventricular mechanics that underpin cardiac function in health and disease. And in 2011, Dr. Shave established the International Primate Heart Project to examine heart disease in great apes and to provide insight into the evolution of the human heart. His work combines both comparative and experimental physiology to further understand structural and functional cardiovascular adaptations to exercise in a wide range of populations, with specific focus on human evolution and the potential for cardiovascular mismatch disease. So I'd like to welcome Rob.
Those are Frank's slides, so let's see if I can find mine. Or I could.
I think it's already open in the PowerPoint below.
There we go. Thank you, Frank. Okay. Thank you for the invitation to be here. It's a real pleasure to be in Guelph to speak to you today. Before I start, I'd like to acknowledge that UBC, where I work, is on the traditional, ancestral and unceded lands of the Syilx Okanagan peoples, and it's a real pleasure to live and work in the Okanagan Valley. It's also a great pleasure to be in a mixed audience, and I think I'm going to be a little bit left field for many of you here, but hopefully I'll spark some interest. Frank, you missed out the heart. Don't worry about it. You've got 15 minutes solely on the heart right now. So we're going to focus there for the next 15 minutes. So I'm going to hopefully introduce you to some of the comparative work I've been doing over the last 10, 15 years. And just with a little bit of an overview of what I want to talk to you about today. Very brief general background. We'll touch upon mammalian ecology. Or ecological niches rather, so thinking about where mammals live and how they live. We'll talk very briefly about the mammalian cardiovascular system, not too much vascular, much more cardiac, and we'll think about human divergence. I'll cover that off hopefully in a couple of slides before really getting to the meat and bones of this talk, which is really a comparison of data that I've collected over the last 10 or so years from the International Primate Heart Project, where I've had the great fortune of working with sanctuaries across the world to be able to collect these data. And then just finally at the end, just some potential relevance and implications. So I'm hoping that I'll get some interesting questions and probably some questions that I've not considered before, so I look forward to that. So before we start, I want to plant a couple of ideas in your mind. So the mammalian cardiovascular system is highly conserved across mammalian species. It's a 4-chambered heart We have a high pressure circuit delivering the blood to the muscles. We have a low pressure circuit bringing blood back to the heart. But despite it being a highly conserved system, of course, mammals live in a variety, an incredible variety of niches. So the hemodynamic challenges that that conserved system has to deal with are quite different. You know, the hemodynamics of a giraffe are very different to the hemodynamics of a sloth. The hemodynamics of a wild dog that's a persistent hunter that runs 40, 50 k's an hour for prolonged periods of time is very different to that of a cheetah who lives in exactly the same environment but whose hunting strategy is 10, 15, 20 seconds. So the hemodynamics that system that is highly conserved across species is remarkably different. And that's kind of the backdrop to some of the work that I want to talk to you about today. Humans. So we've obviously gone through an evolutionary history, and relevant to this talk today, 6 million years ago we split off from our other great ape ancestors, and then really important for this talk is approximately 2 million years ago, 1.8 million years ago, we see the emergence of hunter-gathering strategies. So humans became hunter-gatherers, they moved out from the tree canopy, they moved out onto the savanna, they spent time hunting animals, taking a lot of time to do that, exposed to high temperatures, had to thermoregulate a whole lot better. Yeah, so all of these aspects of the environmental niche, the ecological niche, probably have helped shape the cardiovascular system that all mammals have, but specifically ours in the context of this talk today. So, work from Bramble and Lieberman back in 2004 now, almost 20 years ago, looked at musculoskeletal adaptations within the human. And their underpinning hypothesis here is that humans have evolved to be endurance runners and that they're highly endurance capable. And the signatures in the musculoskeletal system which allow humans to run for prolonged periods of time in a really highly efficient manner. And what that means from a hunting strategy is that they can run at a faster speed for a longer period of time than a lot of their prey species. So in this data, there's comparisons with horses, with antelope, and showing that humans are able to persistent hunt these animals and enjoy great success in providing much needed high quality food, which probably helped our evolutionary history. So that paper published back in 2004, it was written up in or was mentioned highly in a popular science book called Born to Run, which I read back at the day, and it kind of stimulated in me an idea around, if that's the case, then the cardiovascular system has also been adapted and has evolved to manage endurance capability. So this figure will be familiar to anyone who has taken exercise physiology. You can see it in any good high quality exercise phys textbook. But what it's showing here is the remarkable plasticity of the human cardiovascular system in response to exercise. So moving from 5 litres per minute cardiac output sat here up to 25 litres a minute at maximal exercise, maybe even higher than that in elite athletes. So when we mobilise or when we start exercising, we have to mobilise a great deal of blood, and our cardiovascular system has to manage that. We have to shuttle blood away from certain areas and shuttle blood towards the muscle or towards the skin for our metabolic and thermoregulatory demands. So if we have indeed evolved as endurance animals, this system has been selected to be able to do that. That's our working hypothesis, but how on earth did you get at that question? Well, that's where I wrote an email to London Zoo about 13 years ago, and they fortunately invited me to come along, and since then we've done an awful lot of work looking at cardiovascular structure and function in a whole range of great apes, non-human great apes. But the other thing I think it's worth mentioning, and it kind of touches on a number of other areas that we've already spoken about today, is who do we compare to? Do I compare this to everyone sat in this room? Or do I compare it to individuals who are living a traditional lifestyle, who are probably more aligned with what we have evolved to be? And so instead of looking at the weird population that we are, what we try to do is go into populations living a much more traditional lifestyle. So as well as collecting data from the great apes, we also collect data in Mexico with the Tarahumara, who are incredibly well known for their running capabilities. Their tradition, part of their tradition is endurance events that might last 2 or 3 days. And so this kind of activity alongside their subsistence farming mean that they're a really highly active population. So it's not perfect, but it's a much better model of what we have indeed evolved to be. So we go into these communities, we go and work at the sanctuaries where the great apes live in Africa, and we do full cardiovascular assessments. I do some vascular stuff as well, but I'm not going to show any of that today, so sorry about that, Phil. But we are going to concentrate purely on the heart. So this very short video gives you an idea of one of our assessments. So we work in line with the vets who are running health checks on these animals. These animals have been rescued from the bushmeat and the pet trade. They live in sanctuaries, they live in very large enclosures, in range habitat, with structures where they are living in groups that they would normally live with. So this isn't a chimp that is held in a research institute. This is as close to wild as you can possibly get. And so I think that's an important piece. I will also state that none of our work is done unless the animal is undergoing a health check or is being moved. So we piggyback that work. It's important to understand that. So we take cardiac structure and function using ultrasound. We look at vascular measures using ultrasound. We get a host of blood biomarkers to look at cardiovascular risk factors as well. I'm going to focus from a perspective of time today purely on cardiac structure and function, and specifically the left ventricle. So these are data presented, we published back 4 years ago. These are 40 adult male chimps versus 40 adult male Tarahumara individuals. We looked at LV length. The LV of the human is considerably longer. We looked at wall thickness, so the thickness of the wall that we know responds to different types of exercise training. We can see that the walls of the chimpanzee much thicker. Importantly, the relative wall thickness, so the thickness of the wall relative to the cavity dimension, also very sensitive to change in terms of blood pressure, is considerably different with the chimpanzees getting to 0.43, which from a human perspective would be considered certainly on the borderline of disease. And then sphericity, how circular the ventricle is, how round it is. And the chimpanzee heart is considerably rounder than that of the human. So what we have here is an elongated elliptical heart that is more compliant and probably well set up to mobilize blood and to augment stroke volume during periods of time of requirement. The other thing that we noticed, and this is quite interesting because cardiac disease is supposedly the biggest killer of captive apes in the world, and there's groups in the US looking at this. But one of the things we noticed was that in the first few animals we looked at, there seemed to be this highly trabeculated apex. So these folds at the bottom of the left ventricle. Now in humans it's known as non-compaction. It's related to a number of diseases and disorders. And we didn't know whether this is this what's causing sudden cardiac death in some of the chimps? But what we then realized is that it's evident in all of them. So in all of the chimps, they have this highly trabeculated left ventricular apex. So the data here in the bottom right-hand corner is the ratio of compacted to non-compacted tissue at the apex. You can see the yellow lines that we take those measures from. The MRI here is from a chimpanzee who died of a non-cardiac disorder, but we were able to get the heart and put it through the scanner, and you can see as we come down into the apex, you really start to see those trabeculations. They really start jumping out at you. So we know that structure and function are intimately linked, and One of the things that I've spent a lot of time looking at over the years is ventricular mechanics, so how the ventricle actually generates the stroke volume that's required. Now, in humans, we know, and indeed some other mammals, we know that the ventricular arrangement is a helical arrangement, and it moves from a left-handed helices in the epicardium through to a right-handed helices in the endocardium. And what that means is when the myocardium depolarizes, we end up with a ringing motion. The ventricle twists, and so it twists during systole and then it untwists during diastole. Really quite important there. You know, how do we get a 60% ejection fraction when muscle fibers can only contract 15-20%? Well, it's the architecture that allows that. So from a systolic perspective, that twisting, that ringing helps to generate stroke volume. But in diastole, energy that is stored at the end of systole is rapidly recovered, and we get a rapid untwist. And that rapid untwist is really important from diastolic filling perspective. That sets up suction, and it draws blood into the ventricle. Really, really important during exercise. When we've got high heart rates, shorter period for filling, that rapid untwist helps to draw blood into the ventricle and helps to fill the ventricle well, so that we can then augment cardiac output. So that fibre alignment allows us to have this ringing motion in systole and that rapid untwisting in diastole. So given we've got that highly trabeculated apex, what we were then interested in is understanding how that structure might then impact the function of the hearts. So using cardiac ultrasound, we take a slice at the base of the ventricle, base at the top, at the mitral valve, and then at the apex, and we look at how the rotational movement occurs. There's a pointer somewhere. So if we look up at the top here, we can see that the basal rotation between the chimps and the humans really quite similar. So there's the grey shaded area is systole, the white area is diastole. So at the base, the structure quite similar, the function quite similar. When we come down to the apex, however, we can see the standard apical rotation that we see in humans, but in the chimpanzees there appears to be practically no rotation of the apex in the chimpanzees. So what that means is that the diastolic untwist, and this is untwist velocity here, so the speed at which it untwists helps generate that interventricular pressure gradient, and in the chimpanzees, which is the dashed line, they really don't have very much rapid untwist, meaning that they don't generate that suction, meaning that they probably don't fill as well. So from a structural perspective, we think that the apical trabeculations are probably somewhat affecting the function here as well. Now, just because I can show you a difference between the chimpanzees and the humans, it doesn't allow me to tell you that the humans have moved in one direction. It may be that the chimpanzees have moved in another direction from an evolutionary perspective. So what we then have done is we've then gone to the other great apes and used them as an out group here. So these are data collected from sanctuaries around the world. We have a lot of data on chimpanzees. Unfortunately, we don't have as much data from the bonobos and the gorillas and the orangutans. That's simply just there's fewer of them. But when we look at the trabeculation, and forgive me if this is something you're not familiar with, these bullseye plots represent 16 segments of the ventricular wall. So the center one being the apex, the outer ring being the base, and so we are able to look at the ratio of that compacted to non-compacted tissue in each of those 16 segments. So the right-hand figure is humans. We've only got 34 here, but these data basically confirm in what's known in healthy humans. The healthy human heart is really quite compacted, and that ratio is really quite low. However, in all of the other great apes, we see this remarkable trabeculation phenotype. And those are echocardiographic slices at the bottom. You may ask, about the orangutans, why do we not have data on the outer two rings? Well, orangutans have very large air sacs, and because of the air sacs, you can't get good imaging. So you can only get one image, which only allows us to capture the apex. We're not able to capture the other areas, unfortunately. But at that apex, the orangutan seems to be extremely well trabeculated as well. So we looked at that, the relationship between structure and function across the great apes as well. And these data produced by a PhD student of mine, Brian Curry, under review at the moment. These show the ratio of compacted to non-compacted tissue, so that's the kind of degree of trabeculation at the apex. And then these are two of our myocardial mechanics variables, so untwisting velocity, how quickly it untwists during diastole, and then apical rotation, how much the apex twists during systole. And what I think we show quite nicely is that humans are different. We are quite different to the other great apes, suggesting that our cardiac structure and indeed our function have indeed been selected for other reasons. And our belief, well, belief's a horrible word, our hypothesis is that that this is to do with selection for endurance capacity, because having a highly compliant apex that is able to generate that interventricular pressure gradient allows the heart to fill really nicely and generate the additional cardiac output that humans need. So, summarizing those data, in comparison to chimps, human heart is more elongated, has thinner walls and lacks trabeculations. Humans appear to have diverged away from a trabeculated apex to a more compacted phenotype. And alongside those structural adaptations, humans seem to have an enhancement in LV mechanics. I could show you some other mechanics as well. I've chosen to focus on the rotational mechanics, but other markers such as strain and strain rate are also different. And we just wonder whether this is evidence of cardiovascular selection to meet the demands of the human niche. So, why is this relevant? Well, we wonder whether the evolutionary history of humans sets us up for cardiovascular disease when we are living outside of our ecological niche, which most of us currently are. So the working theory is that last common ancestor was well adapted probably for the activities they undertook. So they have thick walls, high degree of trabeculation, they have rounded ventricles, probably fairly good for dealing with high pressures that they see climbing trees and the stress associated living in the societies that they do. Humans, on the other hand, have gone down a different route, and we've become fairly adept endurance beasts. And so we now have an elongated ventricle that's highly compliant, that can generate a higher cardiac output. But often when we see that kind of adaptation, it comes with a trade-off. And our sense is that humans have traded off the ability to deal well with pressures. Now that's fine if we're living the normal life that we have been selected for. But in the post-industrial lifestyle, where we are inactive and we have poor diet, that trade-off may mean that we are incredibly prone to high blood pressure and hypertension and lead to cardiovascular disease. So that's why I'm doing what I'm doing. It seems a little bit out there, but I try and bring it back to what matters to us from a health perspective. So with that, I'd just like to acknowledge many, many people. This work takes a lot of time. We've scanned over 600 animals now, and that is only possible with the help of the sanctuaries and the veterinary teams that we work with, and all of the trainees and colleagues. So with that, thank you very much.
One question. We'll do one.
Okay, quickly. Could you comment on the potential survival benefit of the trabeculation in the primates?
So that is a key question we're trying to answer is, okay, we can understand why the human piece has gone in that direction. Why would a trabeculated phenotype be good for the chimps? I think that it's to do with management of pressure. I think they run at higher blood pressure than we do. and I think they see greater spikes. If you've ever been around a group of chimpanzees, the aggression that occurs very, very frequently must cause massive increases in blood pressure, must see massive spikes in blood pressure. So I think that might be part of it. The other thing is that in the embryonic phase, trabeculations are there, they're the precursors of the Purkinje system. And so as we all develop, they are there and they're important, but I wonder if they may just be residual and they may not actually serve a purpose in the chimps. But by losing them in the humans, we've managed to move to a much more compliant vegetable that's able to form.
Thank you so much. That was really fabulous and really different